And welcome, everyone. We're delighted to have you join us for today's webinar with Edgewise Therapeutics, who'll be sharing the latest results from their clinical trials with zevasemten in patients with Duchenne and Becker muscular dystrophy. I'm Michael Kelly. I'm the Chief Scientific Officer at Cure Duchenne. I'm going to get us started with a bit of housekeeping. First, you'll have an opportunity to ask questions today, and you can click on the Q&A button at the bottom of your screen and submit a question anytime throughout today's presentations. We'll have a moderated Q&A at the end of the webinar, and I'll be joined by my colleague, Dr. Liana Orlando, Cure Duchenne's Vice President of Research. For those questions that we're unable to address, we'll work with the Edgewise team to follow up with you after the webinar. And finally, this webinar will be available on Cure Duchenne's website as a resource for the community if you'd like to share it with others. So let me begin. Edgewise is developing sevasemten, a novel therapeutic designed to protect muscle in both Becker and Duchenne muscular dystrophy. The drug is a first-in-class, fast myosin inhibitor that's designed to protect muscle from the contraction-induced injury, which is a primary driver of irreversible muscle loss and impaired motor function in both Becker and Duchenne muscular dystrophy. This is a mutation-agnostic approach and is expected to offer benefits as a standalone treatment in addition to a combination with standard of care such as steroids, gene therapy, or exon skipping approaches. The company has recruited its global pivotal placebo-controlled phase 3 in Becker muscular dystrophy, and data from that study is expected late next year. And additionally, the company is currently planning to initiate a phase 3 study in Duchenne muscular dystrophy patients in 2026. To talk more about this and the recent results, we've Dr. Joanne Donovan, the Chief Medical Officer at Edgewise, and Dr. Alan Russell, their Chief Scientific Officer, to update us on the latest progress. So without further ado, let me hand it over to Alan, who I believe is going to get us started.
Thank you very much, Mike. As usual, we appreciate your help here getting this going. I'm just going to do the intro before handing it over to our Chief Medical Officer, Joanne Donovan. So as Mike said, I am the Chief Scientific Officer and co-founder of the company. We're a public company, so you get exposed to a slide that looks like this before I start talking. And it's a lot of words just to say that sevasemten, while in development, has not been approved for any indication in any territory, right? So we're just talking about trial results. We hope to continue getting good results, but there's no guarantee of that, of course. Next slide. Thank you so much. Edgewise, we've been around a little while now, since 2017. We're in our 8th year, designing muscle-targeted therapeutics for muscular dystrophies, primarily Becker and Duchenne. Our focus is small molecules that target this muscle injury caused by contraction. So when you use your muscles, if you have Becker or Duchenne, they are a higher likelihood to get injured, and that's our sweet spot. That's what we're hoping to turn down in order to make muscles healthier if you have either of these disorders. And our goal is really to make a once-a-day pill that is mutation agnostic. It's available to everyone with the indication, minimal side effects, and works well with other therapeutics. So it's kind of a real-world solution, if you like, to add in and hopefully improve the outcomes for a whole bunch of people. Fingers crossed. Next slide. Here's the nuts and bolts of what we're talking about. We described some time ago that some muscle fibers are more susceptible to damage when they're used than others, and that's primarily these fast fibers, as Mike mentioned. We also have slow muscle fibers. They're more resistant to injury. The fast ones really take a hammering when they get used. We've made this investigational therapy, sevasemten, designed to protect these fast fibers from damage, and I'll show you just a little data slide on the next one, regardless of mutation, because it's a physical approach. It's not a genetic approach. And clinical trials in Duchenne and Becker are ongoing in order for us to assess what effect this protection has on long-term outcomes in these people. So you've had dystrophin described many times to you, I'm sure. It's often described as a shock absorber. We kind of take a next step just so you can kind of see why sevasemten might be useful. On the left-hand side is muscle contracting in the presence of dystrophin. And dystrophin is present in these complexes on the left-hand side marked by the blue bars. Dystrophin, of course, is inside the muscle fiber, but it effectively links the contractile machinery of muscle, what causes your muscle to contract, to the extracellular matrix, to all of the fibers outside of the muscle, and then across those fibers to the next muscle. So we show it here as a bridge, dystrophin inside the muscle, linking those fibers together. Now, why would that be useful? When your muscle fibers contract, they don't always contract to the same extent. Some fibers might contract more than others. And that linkage helps to support each fiber, such that when one fiber contracts, the other ones are pulled along with it and support that contraction. On the right-hand side, we have an example here of Duchenne where there's no dystrophin at all. Those linkages are broken. And as you can imagine, when those muscle fibers contract individually, now they have no support from the fibers around them, and all of the stress goes along that fiber instead of along and across the whole muscle. That puts excessive stress on your muscle fibers, and that's where this contraction-induced stress and breakdown really originate from. Next slide. Here's the compound in action. So the muscle on the left is a Duchenne muscle from the MDX mouse contracting in a dish. This is a collaboration with really wonderful investigators in University of Michigan. And you can see real time, this is a very small muscle that you can actually shine light all the way through. As that muscle is contracting, those fibers are getting stressed, all of the stress along the fiber, and that's causing them to become excessively kind of active, and that causes them to snap, break, and contract into these little blobs that you see appearing. On the right-hand side, same MDX muscle, but we pre-treated that muscle with a little sevasemten in order to change how that muscle contracts. So now that muscle is contracting, instead of at 100%, is contracting at 85%. We've taken 15% off the top of these specific fast muscle fibers. Thank you for resetting. And you can see that while the muscle is contracting, it is now no longer manifesting that stress response because you've kept the muscle within certain bounds such that it can't get that stress manifesting into the breakdown. So that's our goal in people with muscular dystrophy. We're going to try and turn that down to improve outcomes by reducing contraction-induced injury. At this point, I'm going to hand it over to Joanne, and she'll tell you what we've done on the program to date and how we've made some progress.
Great. Thank you, Alan. And thank you, Mike and Cure Duchenne, for giving us the opportunity to share these data. So we are excited about the Canyon study, which is in Becker. Now, why am I telling you about Becker? Mike told you that the process that you just saw with Alan is the same in Becker muscular dystrophy as it is in Duchenne. It's the severity, the complete loss of dystrophin or the partial loss of dystrophin. So in fact, the mouse that you just saw is the MDX, which is used as a model for Duchenne. So we studied Becker, and that gives us information about how the drug could potentially work in Duchenne as well. So we looked at 69 patients. We included adolescents. And we used as the primary endpoint in this phase 2 study the change in CK, creatine kinase. And when you see those muscle fibers having damage, well, what happens to the membrane is it releases proteins like CK. You see them in the bloodstream. That's how frequently it's diagnosed. And what that is really a marker for muscle injury. So we follow that, and we looked at that early as a marker for, is the drug doing something that we want it to do, like protect the muscle from that damage. So we looked at that averaged over the last half of the study, and we enrolled men and adolescents aged 12 to 50 who had a dystrophin mutation, a Becker course of disease, not on corticosteroids, since that's not standard in Becker, and with a North Star ambulatory assessment that was not quite at the top of the scale. The top of the scale is 34, and not quite at the bottom of the scale, so 5, which is late ambulatory. So a broad range of patients in this study. And what we saw, first of all, on the next slide, from safety point of view, we saw that the drug was well tolerated. The most common related side effects that we saw were dizziness and headache. Particularly the dizziness happened in the first few days, was transient, mild, and actually went away as the drug accumulated. The drug has a long half-life, which means we give it daily, it stays around for a while in the bloodstream, and it takes a while to stabilize the dose. So we got a lot of information from that. Now, what's happening to the biomarkers on the next slide? And what we saw, primary endpoint for the study, was the CK went down 28%. We had seen similar in a previous study compared to placebo. And what a molecule called TNNI2, or fast skeletal muscle troponin, is a specific protein that is in the fast muscle fibers. Now, about half your muscle fat fibers are fast, half are slow. It's the fast ones that are susceptible, and they tell us whether the drug is getting to where it needs to be. So 77% change in fast skeletal muscle troponin. And as a side, that's not cardiac troponin. There's a different one for the cardiac muscle. So strong effect there as well. Now, what's happening to function? So in Becker, the North Star changes typically between 0.9 and 1.7 points per year. And it goes down. That's kind of the blue triangle is the range of natural history studies. And what we saw for the men who were taking, this is the adults who were taking sevasemten, is they were actually stable over a year, and they didn't lose function. So versus the placebo, which was pretty much what you would have expected for natural history. Now, the study wasn't powered for this, but it's encouraging, and it supports us moving, as Mike said, into the phase 3 study that is now fully enrolled, and we anticipate having data next year. Grand Canyon study, it's 18 months. And it is looking at the North Star as a primary endpoint. So this is acceptable to the FDA, looking at a functional endpoint all over the world. And we anticipate seeing data at the end of next year. So that is going to be a very exciting point. Now, why did I tell you all this? Because you were here to listen more, I think, about Duchenne based on the title. But it gives us perspective on what the drug is doing, that the drug is active in reducing biomarkers and potentially stabilizing function. So should I pause here, Mike? Questions, thoughts?
Yep, please do. I just wanted to take a few minutes and alter the format slightly as we discussed, and really just take a minute or two just to discuss what we've seen in Becker. Because as you say, it is really important. And it's important not just because it might guide expectations on what to do with Duchenne, but Becker is a standalone disease. And there is about 12,000 patients that are reported in major markets in the United States, in the 5 major countries in Europe, and in Japan. And there's no treatment options for these. And I believe as we take a step back and look at this, independent of what we're going to learn in a few minutes from the Duchenne study, this is a clinically meaningful result. And the first thing I would say, would you comment on what a reduction in 1 point actually means clinically for these individuals? Because it leans itself into those individuals who can do something versus those who need help in doing something.
Yeah. And even needing help. For example, 1 point on the North Star is it's climbing a step. So can you get up a curb? Or do you need support with a handrail or something like that? Can you get up from a chair without pushing on the arms of the chair? So that speaks to being able to independently use the toilet. So there's a lot of each one of these individual items in the North Star is a functional measure that in everyday life. So again, this isn't a disease that progresses over a year or two. It progresses over 30, 40 years. So we're looking at reducing the slope of decline here and stabilizing the disease so people can hang on to what they have.
Yeah.
Um,
2 points I would add into, Joanne, is that over the 12-month period of observation here that they were on treatment, you followed the line almost perfectly as we'd seen for the decline in function in your placebo group, while you've seen stabilization. And reminding individuals also that the disease is irreversible in Becker, as it is in Duchenne. When you lose that function, it's not easy to bring it back, or maybe impossible to bring it back. So not waiting for a 2, 3, 4 year change where you're seeing a 2, 3, 4 year point decline. Treatment becomes really important here, and it becomes important early.
mhm. Yeah.
Yeah. Can I just to push this a little bit, because I know the company has moved on into an open label extension phase, and these individuals that were on placebo are now on sevasemten, and those that were on the 12-month treatment arm are now on the
Mhm.
extended open label. What have you learned from that? I believe
Yeah.
that's the MESA study.
Yeah, we presented some of those data earlier this year, and when people transition, it gives us the opportunity to look at, say, 18 months. And at 18 months, what you saw here continues. They're still stable over that longer period of time. That is a 36-month study. So we'll have information over the longer term as well, which is important. It's important to patients, obviously. It's important to regulators as well.
Mhm.
And I would add one thing about, people want to know whether they're on placebo or on active drug. And one of the things that the FDA has recently been stressing is they get a lot of information from that extension study. And they don't actually want people to know whether they were on placebo or active. They want us to keep that and not share that immediately, because that data is valuable to them, looking long term and understanding the long term results. So
Very good. I'm delighted to hear that at 18 months on treatment, these individuals with Becker continue to be stable, and that's extremely encouraging. But I also picked up in one of your press releases that the placebo arm, when moved onto drug, stabilized.
mhm.
They didn't continue
Stabilized.
that decline. And I think that's
Exactly.
an important piece of information for individuals
Mhm.
with Becker to understand.
Yeah. Yes. Yes. That you see the same effect. Those ones that came down now are stable. So, because as we move everyone over to the active treatment to sevasemten in the open label study.
Okay, excellent. I understand that the company's met with the FDA earlier this year in a Type C meeting. Is there anything
Mhm.
that can be shared from that as far as guiding what to look for out of the Grand Canyon study that's going to read out next year?
Yeah. So exactly, we wanted to speak to them. We speak to them on a regular basis with meetings to understand, here's what the analysis that we're doing in Grand Canyon. Is this an acceptable? How are we doing this? Is there anything that you're going to want to see differently? And that's the information that we get back from them. That we've got the right endpoint here. We've got what they want to see in the Grand Canyon study. We also compared the natural history data that is in the literature, and we've done an analysis of the natural history data, and how can we use that to compare to the Grand Canyon data, to the data we have. So that is something that's an ongoing discussion with them,
Excellent.
and we'll continue to figure out how best to use those data.
Excellent. I think this has been an important discussion for me because I wanted to disconnect in some ways what's going on in Duchenne and what's going on in Becker. They are two individual opportunities and two massive unmet needs. And the data that's come out on this so far really leans into, excitement that the Becker community needs to feel as we go into the fourth quarter of next year for the top line readout. And
Mhm.
I suspect that 2027 could be a very good year for this drug in Becker muscular dystrophy.
Fingers crossed. Fingers crossed.
Okay, thanks.
So,
I'm going to step back and let you continue on with some Duchenne
good.
discussion.
And I should also add there is a meeting at half a dozen sites across the country later this month at the end, the Becker Education and Engagement Day. So BEED, B-E-E-D. So you can Google that and find out more about that, or on our website, there's also a link to educate people about the disease. And that's organized by a number of nonprofit groups. So what do we want to find out in the Duchenne program? We started studies based on in part, what we had learned in the Becker study. And what we wanted to do was to identify, well, what's the right dose in kids? And to go up in doses and to identify, not to do a phase 3 trial right away, but to figure out what's the best dose for a phase 3 study. So we looked at different groups going up in dose of children, ranging from a dose of 2.5 milligrams to 30 milligrams. And what we saw this in the LINK study, which is a phase 2 study in boys aged 4 to 9. And the Fox study, and the LINK study, they were almost exclusively on steroids. They could be on exon skippers. And in the Fox study, we also looked at children and adolescents who were previously treated with gene therapy. And to understand, we're developing, the field is developing a lot of different approaches. And we see this as an add-on therapy, an independent therapy, but also a therapy that can be used in combination to benefit, even in addition to other treatments that are out there. So what we saw, we looked at biomarkers because we had seen a clear biomarker signal in the Becker study. And we did see that at the higher doses. But what we also saw when we got up in doses, that the boys had some decreases in speed. So we're starting to slow down. We reduced the dose and they went back up again. But that allowed us to kind of identify where the sweet spot is in the dose. This isn't a drug where you just give more and more and more and get more of an effect. You want to be in the right spot where, as Alan showed you, you are allowing function, but still protecting the muscle. So we wanted to find that spot. And we identified that was about 10 milligrams, which is actually the same as the adult dose in the Becker study. And so I'll show you those data, how we got there. It's a complicated study. As I said, there's multiple doses here. And what we also did was, once we saw after 3 months, a placebo-controlled period, we saw that if the biomarkers weren't changing enough, we had them go up in dose. So we looked at a number of doses. We looked at functional endpoints, biomarkers, and most importantly, safety in this early study. 4 to 9-year-olds, ambulatory on a stable dose of steroids. So on the next slide, what we saw was, and we also looked at I'll show you the data on the Fox study, which is basically designed the same way, but with the boys who were on gene therapy. And this could have been boys who had received Elevidys, the Pfizer drug, or the solid drug. And about 2/3 of them had gotten Elevidys. And they were actually out about 4 years at the study start. So they were well past the time where they had gotten gene therapy. These were older boys, age 6 to 14, again, on a stable dose of steroids. Okay, and in terms of safety, what we saw was that the drug was generally well tolerated. We did see at the higher doses, we saw, particularly at 30 milligrams, we saw more side effects. Again, those were reversible. We backed down to a dose of 10 milligrams, which was very well tolerated. And we've kept them on for about a year. They're at least a year and a half, most of them. And I'll show you the data from 18 months. And they've tolerated it well at that lower dose, the 10 milligram dose. In terms of the Fox study, similar side effects. Again, when we saw very quickly when we saw the 30 milligram, there were more side effects. We backed down. We got information from the parents that the boys were slowing down a bit. They could see that. And what we used to look at function in the study on the next slide is we looked at North Star, you're very familiar with, and we also looked at the stride velocity 95th percentile. This is the SIDE device. It's an accelerometer in that it measures how fast boys are going. It's not a step counter. It's a more elegant device that actually the European regulators have approved it to be used as an endpoint in studies. They feel it's very valid for showing change. And they wear this device for 3 weeks, every 3 months during the daytime. And it measures not just step count, but at their fastest, how are the boys running? And you intuitively can see that if they're running faster, they have more function. They are more ambulatory. The disease has progressed less. And there's been a lot of good validation work to understand this. And the hope is this kind of an endpoint can be used more at home. You're looking at what's happening over 3 weeks rather than an hour in the clinic. That is more variability, been traveling the day before. So this is something that is being used more and more in clinical trials. And it appears to be more sensitive than the North Star. And what we found, first I'll tell you on the next slide what our overall conclusion was, and then show you a little bit more about how we got there. And so when we backed down, when we had the boys predominantly on the 10 milligram dose, that was 2 cohorts, about 24 kids. And what is shown here is that at the beginning, they started at doses of 5 or 10. The red zone is they were bumped up to 15, which, as I said, was probably a little too high. It was okay. They tolerated it well, but you could see they slowed down a bit. And the vertical axis is basically how fast they're going. This is the change from the beginning of how fast they were going. The gray line is the natural history data in the literature. So you would expect them to decline over time, much the same way you see the North Star change over time or some of the time function tests. And what we saw after 18 months is they were pretty stable. And that was different than what we would have expected with the natural history. So we are, in terms of trying to find the dose, we are looking at, well, we're a little too high there. And then when we back down, it's in a good place. We're seeing stability. And this is encouraging in terms of being able to go forward to a study where we start from the beginning at 10 milligrams compared to placebo over time, a phase 3 study. Now, on the next slide, we also looked at the other groups. And the group on the extreme left here, cohort 1, was mostly on about a 5 milligram dose. So they did a little bit better than natural history, but not as well as the boys that were on the 10 milligram dose. The boys that were on a dose, cohort 4 of 15 or cohort 5 of 30, we saw them go down at the beginning. In cohort 4, it really wasn't noticeable. It was a small but a measurable decrease. But then when they were shifted back to the 10 milligrams, they go back up again. So we looked at all of this data together to come to the conclusion that 10 milligrams was a good dose going forward. And we've continued all of them on that. Now, what else did we look at? We looked at North Star. And these boys are not on, they weren't boys in placebo. So we measured them compared them to a published model based on their individual baseline function. And for North Star, we saw they were a couple of points better over 18 months than they would have been expected to compared to their matched controls. And the SV95 was considerably better than baseline. So that isn't the kind of that, this isn't, we can't take this to the FDA and say, yep, this is ready for approval. Now we need to do a phase 3 study to be able to go to them for approval. So that's where we are. And we're continuing to treat the boys in the study in Lynx and Fox with a dose of 10 milligrams. And we are, they're an open label, so they know they're on drug. And we're meeting with the FDA and regulators in Europe to get input on the phase 3 design to make sure that they're all on board with that. Who we enroll, what the endpoints that we're using, because in fairness, SV95 is a pretty attractive endpoint to be using in the studies. And we know, others are also interested in doing that. So that's where we are with Duchenne. We've made progress and we're excited to be going on to the next step here. Ask Mike, what questions do you have? It's a complicated story.
It is, but I want to begin by a couple of things just to sort of reframe parts of what you said, Joanne. The development of an NCE, a new chemical entity, is quite difficult. And especially when you've got a new and novel first-time mechanism of action. And taking that into the clinic to show safety and tolerability is one thing, but determining the dose, a go-forward dose, is really the purpose of everything that's been done in here. And this here has been a classical example in my books of how one has gone about trying to take this new chemical entity with a first-in-class mechanism of action to determine what the right dose should be. Too much, it's the old Goldilocks thing. Too much is not where you want to be. Too little is not where you want to be. And it takes real studies, not a single study, but real studies with appropriate biomarkers and readouts in order to get there. And I congratulate you and the team, for what you've been able to do, because it gives you the confidence. And I honestly feel like it gives me the confidence to look at what you're building as far as the phase 3 trial is going to be about. And that there's the foundation that you've built in this. It's complex, and biology is complex, and it's a little bit ugly at times trying to explain it, but that's essentially where we are. And I think that you've now got a really good handle on what the dose should be, what the safety and tolerability looks like in that patient population, and now you'll be able to power it appropriately. And I think the discussion around SV95C, I think, is a really important one, because it's not just an approvable endpoint in Europe. It's actually not just a functional, it is an approvable endpoint in Europe. And that there should guide your discussions, as you start to think about that with the European regulators, and particularly with the FDA. And really, my first question after all of that is, where are you in thinking about where SV95C actually fits into the game plan for the phase 3 and the discussions with the agency?
Yeah. I think it's a very important thing to press them on. And we know that there is a lot of interest in the community for using it to, this is a community that has really borne the brunt of trials that didn't work out, and potentially not picking the right dose to begin with. So we want to, this seems to be a very sensitive way of being able to pick a dose. And we think the FDA should be excited about this kind of approach. As you said, EMA in Europe, the corresponding agency to FDA has approved it they have issued guidance. They haven't approved a drug on that basis yet, but they have issued a guidance that they would approve a drug on that basis. So that's the goal of discussion here.
Yeah, I agree. I think one of the things that stands out to me is that, and this came really in just prepping for the call, how different Becker is from Duchenne. We tend to look at them as a similar sort of disease, but really it's the slowness of the progression that separates them, and that's not the case. They are very different from each other. I think SV95C in your Duchenne study was able to pick up meaningful changes, both in a dose response and in a reversible manner. And it correlated to me really well with where you'd seen the 15 and 30 mg doses really starting to give you some sense of the toxicology or lack of safety, and it fitted with the feedback from parents and from children. So I think you've got a complete picture in there, based on the sensitivity of that assay.
Yeah. That was a really cool thing that you brought up because we heard, within weeks, at the highest dose, the parents saying, I think they're slowing down a bit.
Yep.
And that, we listened to the investigators who were telling us this and went back down again based on what we knew about the mechanism of action. And then to see it validated with the measures was very impressive to see how sensitive the SV95 could be. So it's important to include these things in trials. It helps that trial, but also, the more information we have across trials, the better we can design trials as a community in the future as well.
No, absolutely. Leanna and I had both heard it independently from parents, and so it gives us a sense of where the trial is at that particular time.
Mm-hmm.
And you could see and expect the movement that was going on, as usual, trying to read out and adjust the dose accordingly. One other thing I'll say, I tend to talk too much, so I'll pass it over to Leanna in a sec. But I was particularly intrigued by the Fox trial. If I step back just for a second and remind the community, gene therapy is going to be an important component of where we'll be over the next few years. And microgene was designed in order to transform the phenotype from Duchenne to Becker. Your Becker study really leans into what that may look like if it's powered appropriately and run for a longer period of time. I do expect that combination therapy on top of exon skipping approaches or gene therapy approaches is really going to be an important cornerstone of combination therapy going forward.
Mm-hmm. Yeah.
Yeah. Leanna, any questions on your side that's coming in?
Yeah, sure. I think I'll start with the very basic. I think it speaks to the enthusiasm the community has for these Duchenne trials. So it sort of I have a series of questions which kind of fall into the realm of like, who will be able to participate, right, in the phase 3. And so maybe we could start, if SV95 is going to be the endpoint, right? That's clearly an ambulatory endpoint. And can you speak a little to what your thinking is around for the potential to include non-ambulatory individuals, whether or not they've been on gene therapy or not?
Yeah. Yeah. Great key question, obviously, and that's something we need to discuss with regulators. And sometimes having the tool to answer the question drives who we are able to enroll, certainly. And we think that this would be applicable to the non-ambulatory population. If you are able to protect muscle, whatever muscle you have, it is going to require different endpoints. And that has been hard, basically. We think that it will be ambulatory, and I think that we are trying to get from the natural history studies what the population that will be most sensitive to it. And it likely is going to be boys who are a bit older. The 4 and 5-year-olds are a bit hard because they are getting better. So you've got a mix of kids that are getting better and worse, and that's been that has been the Achilles heel of some of the trials. But this is in flux at this point. We're getting information. We're getting feedback. Both will get regulatory feedback as well as from the experts in the field to understand what the best group of patients. But they would be patients in general on steroids, on ambulatory with North Stars that were not at the very top or not at the very bottom.
Great. And sort of expanding upon that, there's a lot of questions about Duvazat or Givinostat, right?
Yeah.
And from both perspectives, like, are you allowing individuals once they're in open label to be able to add that in, or will future clinical trials also allow for individuals who are taking that drug?
Yeah. At this point with Givinostat, we haven't been allowing boys to be on Givinostat in the trial. And we've run that by our investigators. And what their feedback is, it should work together, but it will not allow you to understand what the effect of sevasemten is. That's the hard part. I think what we have with exon skipping therapy, we ask them to be on a stable dose for a year. And then we stratify them for that. I think when we get to the open label part of the phase 3 and later in the links, we'll be able to allow boys to go on that. So we know that it's got different side effects. So that is also a complication of they're going to be able to be monitored. They have to be monitored closely as well.
Yeah, I think it really speaks to, and I think you guys are being very thoughtful about this. It really speaks to, the benefits of the current situation where we're in, where there's different options, but how that also complicates the landscape, right? To really appreciate and get the data to definitively show, right, without complicating it with other factors.
Yeah.
I think that was it from me, Mike. Did you see other things come
Yeah,
in?
a couple of ones. I see one from my old friend Tushar online, and I know I'm speaking to him later on today, so I'll look forward to that. But Tushar's question is older non-ambulatory individuals and their inclusion in the clinical study.
And there was also a question about contractures as well, which is interesting.
Yeah. Alan, what do you think about non-ambulatory and the drug and the potential use of the drug there?
We got asked that from the very first day before we even had a drug. Would people with lower baseline function still benefit from the drug? I think the answer is yes. It's all a dose response, right? Would they require a different dose? Perhaps. Some of the Becker people that we have in our studies, while they're termed ambulatory, we use quite a large range of North Star, all the way down to 5, which is largely supported ambulation most of their day, right? So we're kind of butting up against that edge in Becker. I think in Duchenne, of course, function has a broader range, and once you become non-ambulant, I think you have to be quite careful. But I think there should be a dose response. Everyone has remaining muscle that needs protecting. So that to me is somewhat separate question, but potentially something we should interrogate.
Yeah, we should actually have that discussion at some point whenever we're together. But I think there's certain diaphragm function that could be preserved in here, and there's ways to measure that benefit in individuals. There's learnings that can come out from the Becker study. But I feel that in parallel with your next year's phase 3, you could have a cohort, to examine both the safety and functionality in older non-ambulant individuals for not just for the label, but really to look for some potential benefits in there.
Yeah.
There was another
in our Becker study. I'm sorry.
Go on, Joanne.
I was saying in the Becker study, we are using the performance of the upper limb and upper limb function as well, because that is compromised certainly in Becker as well. So.
There was another me question from Jennifer. Thank you very much.
You read my mind. I was just about to pick that one up too.
This one is near and dear, Jennifer, to my heart. It's like, why are we not seeing CK move? Right? We definitely have some data from the steroid naive kids that their CK did move. So it always makes you wonder, hey, is that background of steroids influencing specifically that CK signal? Right? This is probably my number one interest, is when we measure CK or troponin or anything in circulation in Duchenne or Becker, what does it really mean, right? And we're doing independent research at Edgewise to hopefully add some color to that so that you folks can understand the data a little bit better, because it's a blunt tool. CK is a mysterious one. You measure activity, not level. And it doesn't always track with the other biomarkers, but I think perhaps we all know steroids reduce CK, right? Then you throw Seva on, maybe it can't reduce it anymore, or maybe it changes how CK is released. lots to work on here to truly understand that CK story, but it is a head scratcher.
Yeah,
And just to
I will
build,
share. Sorry that we were chatting about this before we went live with this webinar. And it's very clear that natural history has changed over the last decade, particularly over the last 5 years. And we're still learning little bits more about some of this here. I think that because of that, I'm really pleased to see changes on tools that are sensitive, like SV95C. And we need more of that really to tease out what is going on, with some of these individuals and not relying on some of these biomarkers.
Mm-hmm. Totally agree with you, Mike. I think we got very excited about the early gene therapy data where that CK dropped through the floor and then bounced back up again.
Mm-hmm.
And then we're all wondering, well, what did that actually tell us? Right.
Exactly.
So there's a lot of information there that I think we still need to lean hard into to truly understand what these things mean.
Yeah. Sorry, Leanna, I jumped on top there, did I?
no, I think we're all saying the same thing. I think, to your point, Alan, right? The steroid naive Duchenne population, you did see reductions, and that would actually be more comparable to what you see in Becker, right? Because those individuals with Becker are not taking steroids. So
No.
the data is kind of a little bit better aligned. And then there's certainly, right, more at play when you have steroids in the mix. And then it's right back to, every other drug that we layer in actually complicates the story. And
It does. Yeah.
Yeah. Yeah.
So any more questions
And
on your side coming up, Leanna?
I see a question about, can they come back in once the LINKS study, if they dropped to start another treatment and for gene therapy? And actually, when we have a separate open label study, that's something that we have talked about as well, of letting those guys back in as well. In terms of Givinostat, we're talking about it's not so much that it not allowing boys that are already on Givinostat into the trial. And that's not unusual in clinical trials to not, to have an exclusion for an approved drug, basically. So.
yeah, I think with that, we basically have covered it.
Okay. Okay, let me wrap it up. So to everybody online and everybody that participated today, thank you for joining the webinar. And a big thanks to the whole team at Edgewise for the work they've done in developing Sevasemten and for sharing this really important update for both Becker and Duchenne for the community. We look forward to future updates as the pivotal trial in Becker is now fully enrolled, and we'll get data on that top line data in the end of next year. We'll get in touch with all the attendees to let you know when the webinar's online, and and thank you for everybody else that's joined us. Have a wonderful rest of your day.
Thanks.